Okay, sir.
Okay. Yeah. So, hello, sir. The very first question is, I have seen a lot of Indian scientists who are life scientists have their PhDs after their BScs and MSDs, but you have done the PhD after the MBBS. What was your mindset back then? I mean, that was way beyond when research was, actually interested or kept in terms when, as a medical professionals.
Yeah, so the reality is that if you're in medicine, then the only sort of path or the career forward would be to do an MD or an MS and then do a DM or an MCH and then do clinical practice. So at the point in time when so we sort of finished MBBS, there was no real career path in areas which are not what you call formal departments in medical colleges. So when I say formal departments in medical colleges, I need to qualify this statement because in India, the medical system is largely fragmented. So there are medical colleges which are in the state, which run formal degree programs in medical surgical specialties. And then, of course, there are institutions which are under the Act of the Parliament, the typical of the likes of AIMS, PGI, so on and so forth, where they have much more freedom to create departments. And then they would have some additional super specialty departments that they have created. But beyond that, in a very emerging or a new area, very unlikely that you will find a department or a formal program in medical education. And then, therefore, the only option to pursue what you were interested in was to probably shift gears and move into a non sort of medical institution where you can acquire advanced qualifications. So the real reason is that at that point in time, and unfortunately even at this point in time, we don't have probably an MD program in say clinical genomics or an MD program in genomics. So that essentially is a problem. And in many ways, the medical education system is quite rigid in accepting new departments and new fields.
Thank you very much for this question, sir. But even after you joining the clinical genomics branch, I do not see a clinical genomics branch even 20 years. And I am not very sure that in the next 10 years, we could see a genomics MD branch. Do you think that
Yeah.
is possible, sir?
So theoretically, that is possible as long as you have a large number of clinicians who have trained in the field come up, who have formal degrees in the field come up. They should be able to start new departments. So it all boils down to a large number of people who can seed a particular department. So you cannot really start a department without people, right? See, most of the institutes can actually start a human genomics or clinical genomics department, but then the problem is where are the people? Right? So unless you have quite a large number of clinically qualified people, you will not be able to start a department. So it's a chicken and egg story, right? Unless you have a department, you don't have people, and if you don't have people, you cannot start a department.
Yes, sir. Sir, but the branch that you have chosen, that is genomics, it is completely a, what do we say, bench research, not the clinical research. So what were the challenges you faced during your journey from a complete clinical environment to a bench-based research environment? I mean, how did you look into it?
Yeah, so it was a conscious decision at that point in time that I had to leave clinical medicine if I were to pursue what I wanted to pursue. So yeah, frankly speaking, what I was really interested at that point of time was to actually do things at the interface of clinical medicine and computer science. And genomics just happened to be also at the right point in time because just a year or so before that, the human genome was announced, and there was a lot of interest all across in trying to analyze genomic data. Now, by having said that, it is not that genomics is a non-clinical field. Yeah, my PhD was not on a clinical topic altogether. I used to work on viruses and microRNAs that can target viruses. But quickly after I started my lab, I started focusing back into looking at application of genomics in clinical settings. And so one of the largest programs that we run today is actually looking at clinical samples and patients with extremely rare genetic disorders and trying to use genomics to provide them a diagnosis and in many cases also provide them the right treatment. So it is not really something that you can call as non-clinical. Yeah, the only difference is that I don't see patients in a clinic, but I do see patients on the computer in terms of the clinical data and the genomic information.
So basically, you tend to say that the graphs and the charts that are present are more clinical rather than, I mean, by sitting on the bench, you would want to see all the clinical data in a chart rather than directly going and visiting the patient.
Absolutely right. I mean, on almost all of my PhD, and in fact, from 2006 until today, I have not stepped into a wet lab. So practically all that I do is on the computer. So I don't really miss seeing patients, but yeah, it's a way that you can sort of think. It is not that somebody who does biochemistry or somebody who does pathology is less clinician, and probably, I mean, the best clinicians I've seen in my entire life is probably a pathologist.
Yes, sir. Sir, uh, my experience, I easily get fascinated by many things in medicine. But out of all those things, how do you come to a one single thing? Like, there are many topics that you were interested, but what made you fascinated on clinical genomics?
Yeah. So
And,
the
yes,
factual
sir.
reason for that is right. So in life, you cannot predict everything that you want to have, and you cannot predict where you will end up in. Right. The best option is to actually keep your eyes and ears open and pursue your interest. And my interest generally was essentially using computers in medicine. Yeah, so I'm doing that. So I don't have bad feelings about what I choose. Genomics is one opportunity where you can use that, but of course there are many more opportunities where you can use computers in clinical medicine. So I mean, it just happened. So it is not that I planned to become a genomic scientist, and when I got out of PhD, I wanted to start working on rare genetic diseases. No, it just happened by chance.
Sir, how come a student like like me or like any other medical student should decide his research topic, or this is the area that, let's say that I am interested in research and I want to work on research, but how should I choose my area of interest? What are the prerequisites, or is there no prerequisites
I
at all?
I think you should not choose your area of research in a very narrow field. Um, I see that quite often to people whom I talk. This is also the PhD season where we take in a lot of PhD students, and a lot of people come with very focused notions about what they want to work on or what they want to pursue, and in many ways those are in some ways unrealistic. Right? You need to have a broad idea of what is a general area that you want to work on. Right? What you need to specialize on probably comes to you by chance, by interest, by exploring things. So a lot of such preconceived notions of what field I want to work on is many ways based on very little what you call logic, thought, or actual experience. And I think what you should really do is to experience what the environment is and then decide what you want to work on.
Okay, so does that mean that you are actually suggesting students to take a 6 months or a 3 months break and work on a research environment and then
Absolutely.
decide? Oh,
Absolutely.
so do you think that it is feasible in Indian lab setups and in these things?
See, in this world, everything is feasible provided you put your efforts in the right direction, right? So I don't think you will be thrown away from any lab if you approach a lab saying that I have a 2 months holiday after my exams. Can I come and spend time in your lab? I don't think any PI would say no to you.
Okay, so but let us say that I want to go and work with a PI for 2 months. But what are the basic things that a PI is expecting of a student to at least know?
See, so anything that is less than 6 months, no PI expects anything from the student, right? In
Okay.
in science, 6 months is a very short time, right? and especially when you are from a medical background, it's very well understood that you would not have experimental skills. In many ways, you would not have computational skills. It's very well understood that you have just come to explore what is happening around. So you will get a very small project or a program that you can probably interact with others in the lab, sort of learn how it happens. It's not really to impart a set of very specific skills or give you a certification or anything like that. It's just understood that you want to come, understand, explore, and then make your decisions.
Okay,
Right?
sir.
So it's not that any PI would have an expectation that you will publish a paper, and actually you should not have such an expectation, even from the student, that I will come for 2 months and I will have a paper. I mean, that's not possible.
Sir, but I see right now I'm seeing a lot of students focusing on more research projects. But I do understand that if we focus on an observational studies, maybe it is possible to do a paper in 2 months. But is it really possible to do studies in experimental research, like I mean, in a short amount of time? Is the output going to be a
See, the real big problem here is most of the students would want to do research. I'm sorry to say this, I've seen this over and over again. Most of the students are interested in doing research because they want to have a publication that they want to apply abroad. I mean, let's be very plain simple about it. Most PIs know this, and most PIs would not want that to happen because it takes a lot of soul stress on themselves. You cannot have just a student come in and say that I want to have a paper. I mean, that's not really possible. Let's be really realistic. The entire purpose of doing research is to be able to explore something new, find something which is unknown, right? Dig up something that people have buried long before, right? And re-examine it. And that really requires a different kind of a mindset and different kind of patience. And it is not all about publications, right? It is also about fun of realizing something or fun of finding something new. And yeah, so if you really approach somebody to say that I'm interested in something, I want to explore something, most PIs would say yes, right? But if you approach saying that I would want to spend 6 months and I would like to write a paper, then most people know that, okay, this is not real interest, but means to something else. So if you are really interested, if you're really passionate about what you want to do, I think most labs are pretty much open.
Thank you for
Sir,
clarifying that, sir. Does
I
having no
want to
research
add,
experience? Yes, sir.
sir, I completely agree with you, sir. The number of medical students choosing the medical research as a career was very low. But I think few of the students would be motivated after this session so that they may choose their career, medical research as a career in future. So could you add 2 points on this, like choose career and your journey of becoming the scientist?
Yeah, so I passed out of MBBS in 2003 from Medical College, Calicut. I'm not sure if there are anybody from Calicut Medical College. Yeah, this is probably an old medical college. We were the 42nd batch. And yeah, just after MBBS, even during MBBS, I was sort of, it's some sort of self-realization that probably your best interest is not really doing clinical medicine. So most of the time I would probably spend my time in the library trying to look at a lot of articles, journals, books that others would not typically read. I'm sure most of people who have ever been in Calicut Medical College would appreciate that the library has actually 2 sections. One section for most of the latest clinical books that is very busy, and it has a very large collection of old books, right, from Advances in Genetics, Nature, Genes, a very old collection. I will say very old collection dating from 1950s, 1960s, and so on. Very well maintained, bound editions of a lot of journals. And that was like a free area because nobody would ever dare to get into that space. I got really fascinated by a lot of these things, and there was a fantastic librarian who would help me around. He would really go spend his time and effort to pick up a lot of journals that I wanted, very unlike most other medical colleges. And I practically spent a lot of time in that section of the library because I loved reading a lot of things that, so this includes a lot of things about embryology, a lot of initial experiments on how microscopy was used to decipher a lot of ultrastructures in the embryo within cells, a bit of karyotyping. And I still remember one of these days, the librarian, when I was stepping into the librarian told there's this new book that has come. I've kept it for you. And the book was called Genes. I believe the version was Gene 6. This is by Levin. Very well read book. So the next over, I would say 3 months, I sort of finished that book from cover to cover. So it was so fascinating reading that book because it explains basics of genetics and genomics in very, what I call simple language with a lot of good illustrations. And that really got me fascinated. And that is also the time when the human genome got announced. At least the chromosomes started getting announced. And then, of course, after passing out, there was a 1-year internship. So I sort of scraped through the internship. Most of the clinical specialties didn't really interest me. What really interested me in the internship was one, psychiatry. I don't know, for some reason. And of course, radiology, because we had a 15-day radiology posting. And we used to have a very fantastic professor in radiology who would go back and explain a lot of things on CTs. I mean, CTs and MRIs were really new at that point. So then I finished my MBBS. I decided that I will need to at least work for some time to get a feel of how it is. So 3 months I worked in a local hospital as a resident, and then I decided to apply for a PhD. And there are very few institutions which would take an MBBS student for a PhD. So my first application was to one of the major medical institutions. I would not want to name it. I went and met a very famous professor in there, and he told you should not probably do a PhD. You should probably do an MD and then come back. So then I applied to a couple of other places, one IIT and IGIB, where the first PhD batch was open. So I applied, I came for an interview, and after the interview, I was sort of confident that I might really get through. And I think a couple of weeks later, I got a call saying that you've been selected. Why don't you want to join? So that's how I joined.
Thank you so much, sir, for sharing that journey with us, sir. But when you said that a very good professor of yours, I mean, when he said no, what was your feeling at that time of you should go and do MD?
So yeah, so there are very number of things that has come which made my decision on what to pursue and what not to pursue. And these major decisions or time points in life are not really planned. This just happened by chance. That's how I want to believe it. During my 3rd year, I believe I did a project with one of my pathology professors, quite a well-known person, Professor Aravindan. And I used to work on a very simple but elegant kind of an observational study. And the idea was he had a series of papers at that point in time where he had looked at pathological samples of appendicitis, the excised samples. And what he found was there were a lot of eosinophils in the appendix. So the idea or the hypothesis at that point of time was probably it is something to do with allergy. And if there was allergy, then there is something to do with the food that has been taken in the previous 3 to 4 days. So we went to the clinic surveying people who had recent appendectomies, trying to ask questions on what did they have in the last 72 to 48 hours before the onset of the pain. And it's a quite simple questionnaire that we sort of put together. I believe we put together close to 60 to 70 patients in 2 or 3 months. And then we sat down together to analyze the data, looking at the statistical methods. And I learned a lot of things from that. And that really excited me because I was otherwise also very excited at pathology. Then, of course, there was another good professor in the community medicine department who taught a lot of research methodology. And he used to spend his time after 5 o'clock in his office with a bunch of students, and he would teach a lot of research methodology on how to ask a question, how to frame the right question, how to go about doing the study, how to plan a study, how to take care of the confounders even before we could look at the study, or even before you have planned the study. I learned a lot of things by just interacting with this particular professor, and he was very passionate, and he would practically spend time until 9-10 o'clock until we go for dinner, and we used to have dinner together most times, and then I would leave for home after 10 o'clock. And this used to be a quite regular sort of, and this is very informal. It's not like it's a very structured course or anything like that. A bunch of students who were interested would come to his office, and everybody expects him to be around. He would finish his job, he would give us something to read, and then we would keep discussing, and random things that we would keep discussing. So that was 2. And the third was essentially my PhD mentor. So I still remember this. So in the Institute IIIT-D, we typically take in around 12 to 15 students every year. And then each of the students would go and meet around prospective faculty members, and then randomly discuss what their ideas are. And sort of, it's what we call as a speed dating. Over a week, they meet each other, and then they sort of fix, who joins which lab and so on and so forth. A tradition that still continues to date. So I met everybody, and I didn't pretty much, get a feel of, or a real feel of what I need to work on. I really wanted to work on computers, but I didn't find anybody really interested in working on computers. So this is almost at the end of the week. I was really dejected that I didn't find somebody who really wanted to work on what I wanted to work on. And but the other faculty told Missy, you should not leave. You should meet the director and then only leave. And he was not in town during that days. So in I believe it was September 28th, and the director was around. So after, so it's also a foundation day for the institute. So after the program, I went, and so he was out there actually smoking. So I went and met him and told, say, I'm so and so. I wanted to work on this, and I think I didn't find anybody, so I just wanted to meet you and say hi before I leave. And what he said was, yeah, no problem. So you wanted to work on this, right? I was in your interview, so fine. You work on what you want. You join my lab and you work on what you want. And I was really taken aback. And how can somebody say that you work on what you want and I just join my lab? So I decided, okay, let me try. And just it was really good because he rarely interfered in what I did. He really promoted me to work with a lot of other people in the institute. So practically during my PhD, I have papers with at least 6 faculty members. And we worked on a lot of problems, starting from Mycobacterium tuberculosis, then working on microRNAs, then working on DNA, quadruplexes. We worked on quite a lot of stuff. I mean, not just one thing. And that was really something that turned around because that got me interested in a wide variety of stuff. It educated me on a lot of things that I never knew was possible. So I think all these opportunities come in if you are open to exploring opportunities. And this was really random occurrences. I don't think this is planned by anybody or really fixed in that way. So if I had decided and joined, say I want to work on cancer, probably I would have joined a lab working on cancer, but then probably I would not have explored a lot of other things or would not have been able to work with a lot of other people. So yeah, that's in short how things happened. It was probably not planned, but yeah, it happened for the good.
Sir, what are the career opportunities after PhD?
So for most medical personnel who would want to pursue research, there are two things that you need to keep in mind. And this is what typically I advise students who come for advice. So if you want to pursue clinical medicine, right, or if you think that you want to be a clinician first, right, then always finish your education before you think of doing research. You cannot be best of both, right? You can be a good clinician and a good researcher, or you can be a former doctor I call myself a former doctor and a researcher. But very unlikely you can become the best of both because there are a set of skill sets that you require to do both. So if you ever want to see patients in your life, what I think is you should finish your education, you get your MD, DM, whatever, right? And then pursue research. And there are a lot of opportunities to pursue research as clinicians. But if you think that I don't want to see patients anymore, I want to really work upon a topic which is either pure or applied, but I would not at any point of time sit in the clinics, then probably the best option is to shift after MBBS and do a PhD. There are a third set of people who would finish their MD or MS, right, and would not have a specialty in that particular area. Right? They would still want to see patients, but they want to see patients in a very super specialist area for which a department doesn't exist. Then, of course, there are options where they can do a PhD after their MD, right? Which is the regular route of doing a PhD. Typically, most universities, you can do a PhD after doing your MD. So there are three options, and largely depends upon what you feel about your future career. So if you ever want to see clinical patients, then you should finish your regular clinical degree before you end up in a formal research degree, if you want a formal research degree. But that doesn't say that you should not do research at any point of time. You can do research at any point of time, right? But in terms of dedicating time for very specific set of skills, the timing would change depending upon what your interests are.
Yes. Sir, how to frame a right research question, sir?
Pardon me?
How to frame a right research question?
See, there is nothing like a right research question, right? They are only research questions which can be proven or disproved. Right? And depending upon the tools, techniques, and the data that at some point of time you can acquire. So there's nothing called the right research question. There's no perfect research question, right? They are only good research questions and bad research questions. Right? There's nothing called a perfect research question. And there is no simple rule that you do this and you get a good research question. Am I audible?
Yes, sir. Yes, sir.
Yeah. So there is nothing like a fantastic way of framing a research question. It actually happens by a lot of discussion and a lot of experience by framing questions.
Yes.
Sir, thank you so much, sir. And can you add 2 to 3 points on clinical research, like it was developing nowadays? So
clinical research is what I call as research done by clinicians to address very pertinent questions that they see in clinics. And this, I could largely classify this into two types. One is a type that you sit in the clinic, you see a set of patients, you have a few observations, right? To make based on the clinical outcomes of patients or the progression of the patients. And then you ask a question, what is so relevant, important, or rather ask this question, why is it happening? Right? That's one kind of questions which are motivated by observations, right? Which are motivated by peculiarities in the patients that you see, and you pursue digging deeper into addressing why does it happen. The other kind of question that you typically ask is the kind of clinical research where you look at outcomes of treatments, right? And ask this question, if I give this drug versus that drug, what does happen? And that's a typical, what you call clinical research that involves clinical trials and a gamut of things around that. Now, there's a small, minor proportion of researchers who ask very basic questions, irrespective of what they see in clinics, and then try to apply that back into clinics. But that's a very, very small proportion, at least in India, that's a very small proportion of clinicians who do that. Most of the clinicians that you see across who do research can be classified in the first two category of individuals. People who are either motivated by the clinical experience or clinical observations, or people who try to implement findings from elsewhere into clinical settings to look at the progression of a disease, the utility of a particular drug, so on and so forth.
Okay, sir. Sir, and we have a few questions, sir. So I think we can go for questions now. So most of the people are asking, like, is it good to do research in MBBS UG, and in which year is it good to start the research?
So yeah, there's nothing bad by doing research in your UG. It doesn't hurt you, but don't do anything because everybody else does it, right? You do something because you like doing it or you're passionate about doing it. And that is true with everything that you do. I mean, even if you do clinical medicine, do it because you're passionate about doing it, not because 10 other people in your batch does it, and therefore I should also do it. I mean, and that really hurts you, because then you will end up into comparison, and then you will always be depressed that somebody else does better or worse. I mean, why get into the comparison? You are yourself, right? If you like doing something, you do it. It doesn't hurt you anyway. And probably the best time to do your research is just after your first year, because practically after the first year, you don't have exams at least for 1.5 years. You would immediately start your paraclinical topics, which is pathology, pharmacology, and also start your clinical postings. So that's the right time, because you have a lot of time, free time. You don't have exams for 1.5 years, and you have a lot of knowledge from the basics, which is anatomy, physiology, biochemistry. A lot of skills or a lot of observations that you can do in the clinics. So you have some inroads into the clinical setting, and you also have some basic knowledge about the pathology and the pharmacology, so that you can sort of start framing very interesting questions. So I think that's a right time to start doing something if you're really interested in doing, because later on you get busy with exams, with clinical postings. At least in the final year, I think most people are really afraid of about the exams and how to go about cracking the exams.
Yes, sir. Yes. Hello. Yeah, continue.
Sir, after MBBS or after doing PG, what institutes can we apply, sir? What are the institutes available for research, like IITs and something? Can you tell us some list of institutes?
Yeah, so there are very few institutes which would take in students just after MBBS, right? This broadly includes all the institutes which are set by the Act of the Parliament. So typically this would include all AIMS. This would include all IITs. This would include JIPMER. This would include PGI. And then apex institutes, like for example the ICERs, national laboratories, are typically the people who would take you just after MBBS. most state universities would not take you after MBBS. There are very few exceptions of that, but most state universities would take you only after you finish your MD.
Sir, how much salary can we expect, sir?
Yeah, so you should not expect what you get. You should probably expect half of what you should get otherwise as a senior resident in a hospital. So don't come to research because it is well paid, right? You will never match the salary of a resident if you're doing PhD. Never.
Just about career security, I'm asking.
See, if you're looking at career security, then I would say pursue clinical medicine. Right, that is probably more secure than doing research. Because I'm not saying there's no career security in research, but practically in research, you're always chasing a dream. And to chase a dream, you need to have grants. So you need to chase grants. To get grants, you need to have publications. You need to have patents, depending upon how you want to quantitate it. So there are quantifiable measures that you need to pursue. So there is a lot of other variables that you need to address all towards when you do research. So yeah, if you want to have a very peaceful life, very well set out chart where your day is fixed from morning to evening, then probably research is not probably the right place for you, right? In research, everything is variable, everything is flexible. An experiment that you planned might not work. A reagent that you thought was in the lab is probably not in the lab, and you might need to wait for 6 months. So there are a lot of variables. A grant that you thought would fund your passion probably was rejected. The paper that you uploaded yesterday came back today morning. So yeah, if you want to have a very set, planned life and career, probably research is probably not a good idea, right? But if you really like doing what you want to do despite all the odds, then yes, probably you could go to research.
So may I also, I guess most of the Indian students find it very difficult about the pathways and what are the various opportunities available for medical students to pursue research. After MBBS, what can a medical student go for? Like recently we came across some institutes also provide MSc in cancer cell biology. So what are the other opportunities one MBBS student can go for in India or abroad?
Okay, so I would still restate the first thing, that if you are interested in clinical medicine or you see yourself wanting to see patients in clinics in your life, right? Then first you need to pursue your regular medical degree, whether it's an MD or whether it's an MS. You need to pursue that for sure, because in India, for example, you cannot practice in a medical college, right? You cannot, for example, go in a medical college and join a medical college faculty without the accredited degree programs that you otherwise have, which is an MD and a DM program, depending upon whether it's a specialty or a super specialty. Now, that is something that you need to keep in mind, that if you ever see yourself in a medical college seeing patients in your life, then you need to pursue your regular degree programs before you do anything, right? Because each of these degree programs, you need to crack the NEET, right? You need to get a good score, you need to get trained through the rigor, and then only you get accredited to do what you want to do. Now, having said that, now there's a second set of people who want to say that I don't want to see patients anymore, right? I'm perfectly fine not seeing patients anymore. I will probably not be a faculty in a medical college at any point of time. And if you're fine with that, then, and you want to pursue research, then it's a detour from clinical medicine to pursue research, right? With a clear understanding that you don't want to be the faculty of your medicine, surgery, or for that matter, any super specialty departments. at any point in time, right? Then there are multiple opportunities that are open. The opportunity number one is if you are interested in pursuing a 2-year or a 3-year program to build skills, then there is a very, especially in that interface of engineering, there's a very good program run by IIT Kharagpur, which is called the Masters in Medical Science and Technology. It is mostly a technology-oriented program where you get a master's degree with a lot of exposure into engineering and chemical sciences. So if you are wanting to work at that interface, mostly in the area of biomedical devices, biomedical technology, then that is a fantastic program where a degree is offered, a master's degree is offered to MBBS students. They have a very small intake annually, but this is a fantastic program. If you are very sure that you want to pursue research in a particular area, for example, you're very good at computers, you want to pursue informatics, or you want to really pursue cell biology, then probably might not be a good option wasting time in a master's degree. Probably joining a good lab which matches your wavelength and your interest directly after MBBS might be a better option, right? But having said that, there are many more options available and open for clinicians who are either MD, MS, or after their DM or MCH programs to pursue research. And there are a lot of funding opportunities and training programs available where you can get into research, even quite a large amount of time dedicating into research after you have finished your medical degree programs. So most of you, I believe, would want to be in clinical medicine at any point of time. So the best option would be to pursue full-time research or a significant amount of research after you finish your regular degree or postgraduate programs. For the minority who
being a second year student, no, sir. So being a second year student, if I have to proceed with research papers, what are the procedures to be followed, sir? Legal procedures, and how I have to approach with that research and how I have to submit it. Can you explain about the legal procedures, sir?
So typically, when you say writing a research paper, there are two parts of writing a research paper. One is the idea that goes into the paper, or that's a hypothesis that you sort of frame. And second is the data. And the data could be clinical data, the data could be public data, the data could be what you call analysis of data that's already available. So that depends upon what you want to work on. If you were to work on a clinical data, of course, you will need to get the clinical approvals, which is ethical committee approvals of the institution to pursue that particular piece of research. And in most medical colleges, to do that, you will need to also have a supervisor in the relevant clinical specialty who would guide you appropriately on collecting the clinical information in a very systematic way. In most ways, unless you are completely equipped to do the analysis, right, you typically would choose a mentor or a professor whom you are very comfortable with to be able to put together the analysis and be able to write the publication. So, but having said that, there are also a lot of places where you would not require a mentor to, for example, publish a paper. And that is perfectly fine. Like, for example, if there is a genomic data that is available, you're quite educated and good at analyzing genomic data, for example, then you don't need anybody's help to be able to publish a paper. And neither would you require an ethical clearance because you are essentially working on data that is otherwise publicly available. So that's in basics. So if you were to work on a clinical data that needs data collection, then of course you need to get ethical clearance. If you need help with collecting, assimilating analysis of data, then of course you need to have a mentor whom you can work with. But of course, in a minority of cases, you would be on your own, and that doesn't require or that doesn't fall in any purview of ethical or legal restrictions. You can of course work on what you like to work on. There's nothing illegal about it.
So what are some of the good books and online resources to learn about biomedical research for beginners?
See, there's nothing like a book for research. right? They're only publications and books that will get you interested in a particular area, right? And this could be any area. This could be clinical medicine. This could be basic research. This could be anything. This could be analysis, statistics. So if you're interested in some area, of course, you will find more and more or read more and more about it. There is nothing like a dummy's guide to doing research, right? because you cannot build one to do research.
Sir, can two different, like can an MBBS and a BHMS student can collaborate on a paper and try to do something like integrative medicine concepts?
Anybody can collaborate with anybody as long as there is interest between two people.
Okay, so there are a lot of questions on. I mean, do you have any idea apart from ICMR STS? Is there any research opportunities for the undergraduate students to do while they are still in the college?
Yeah, so for MBBS students, what is very popular is the STS program, which is funded by ICMR to do research. But having said that, what you should also keep in mind is that most research doesn't require a lot of money, nor does require funding. What really requires to do research is good ideas and good ways of collecting and analyzing data. Right? So in practical senses, most of the research that you would want to do would not cost anything, and therefore you would not require a fund. Coming back to the same point, that what you really require is a good question, a good hypothesis which can be addressed, a good systematic way of data collection, and systematic way of data analysis. Nothing else. Now ICMR has started a new program. This is for people who have just passed out of their MBBS but want to pursue full-time research leading to a PhD program. ICMR has a very recent program which gives you quite a generous fellowship, close to ₹1,00,000 a month for 3 years for you to pursue research. And the idea of doing this, of course, there are very limited numbers. The idea of doing this is to actually help the people who have decided that they are not going into clinical medicine, but actually going to do 100% of research. Right? So people who want to do MBBS and then pursue a PhD just after MBBS, this is the right kind of a fellowship, and typically you need to apply it within 1 year, within 1 year of finishing your MBBS.
Thank you so much, sir. Sir, how do you compare research in India and research in abroad? So can MBBS students actually apply for research in abroad?
Yeah, you can apply research abroad because MBBS program in India is a 4.5 plus internship. So yeah, you end up having 5.5 years almost of teaching plus experience. And is considered equivalent to a 4-year program. So you would be equivalent to any BTech program while applying to US universities, which can take you in directly for a PhD program. So you can apply to most European or US universities just after your MBBS if you were to pursue a graduate program or a PhD program.
So to apply to those graduate or PhD programs, I think we should have a little bit of a research background in our CV to show up. So doing some observational studies or these kind of studies, does they have any importance? Or
see, one thing that you need to keep in mind always is that never build your CV to apply somewhere, right? You will be very disappointed, and you will always be disappointed. Never build your CV to apply somewhere, right? Build your CV based on what your passion is. Right?
Yes, sir.
And there are very few things that most people look around in a CV, and clearly they are not publications. There are many more things that a person looks in a particular CV to assess whether it's a good candidate or a person who has created a CV to apply for a position. Right? It is very evident. Right? So never build your CV, right, to apply for a position, because people have seen hundreds of such CVs who have been built to apply for positions, right? And they can really, really catch it. So don't think you can do that. But if somebody is really passionate about something, right, have gone out of the way to pursue what he or she wants to pursue, right, there are other things that you look in the CV, right, and typically those are the things that really make a difference.
If I may ask, what do you see in a CV, sir?
I don't read CVs at all. Frankly speaking, when I hire a student, I don't read a CV because all CVs are wrong, right?
So
But for example, if somebody were to apply to me, right, I worked in the area of computational biology. So if somebody, a medical student, applies to me saying that, see, I like computers, I know programming, right, and during my entire course, I had a lot of free time, and these are the things that I have built. Right, this could be websites, this could be some software which is on GitHub, which is verifiable, so somebody can open up and see. Somebody who says that I've created this fantastic app that 10 or 100 other people have used, right? That means that that person is passionate, right? But if somebody comes and says that, yes, I have attended 10 conferences and have certificates to it, right? I clearly know that, yeah, this is good for applying somewhere else. Nothing wrong with that. I'm not saying that applying abroad is a good thing or a bad thing. I mean, if people have their own choices, and we should really appreciate their choices, right? The only question is whether whether it is going to be mutually beneficial to somebody or not, right? But if people cook up CVs, and very easy to actually see through that you have made a CV to apply something.
Okay, sir. Thank you so much, sir. So you actually see the passion in the student.
Absolutely, that's all what matters in science.
Okay. So I would ask all the students actually to look into that if you are applying for lab.
Yeah. So if you're not passionate about science, you should not do research. I mean, that's what I would say. Right? Don't do research because you want to have a paper to apply somewhere. I mean, that's. I mean, you'll always be unhappy about it.
Sir, Madhav sir, we have few like many questions from the audience, so we should complete it as fast as possible and wrap up the session. Sir, one guy has done his ICMR-STS research project, sir. He is asking that can he publish it by self without his guide?
If the person has done the analysis, data collection with a guide, then I don't see a reason why he should publish without a guide.
Okay, sir. And sir, for another girl, she has done a research project ICMR-STS. She is saying that her guide was published without her name. So what does it affect her research career like that? She is asking.
See, one thing that you need to keep in mind is that world is not fair. Like there are thieves on the road, right? There are thieves in every profession, right? And you should realize that, and you should handle that. Now, in many cases, you will not be able to confront a person and be able to fight with it. So you can decide whether you want to fight with it or not fight with it. But of course, if the data was collected by you and you have a lot of contributions into the formulation of the idea and the data collection and analysis, of course, you could go back to the institutional committee, right, and raise a complaint. And that's a perfect legal way of doing it. All institutions have redressal forums for research integrity. So that's what you can do. But one thing that you need to keep in mind is that one paper is not going to make or break your career, right? Your career is going to be built upon passion, right? And if you have passion, there are 100 more ideas that you can work on, and you can build your passion. Now, one paper, an authorship here or there is not really going to make any difference. And you should not really be unhappy. You should not be really unhappy, dejected about something happening. In science, yeah, you will see a lot of more places where you might get dejected. So yeah, it should not really affect you. You should really pursue what you want to do.
Okay, sir. Thank you, sir. Pawan Kumar, you can ask your question now.
Sir, sir, as you said that second year is the right time to do research for UGs. I have small doubts, sir. Without knowing some minimum knowledge on MBBS subjects, is it right time, sir? Can you tell me any advantages of doing in second year?
See, the second year is the right time only because I said that you would have a lot of time, right? Unlike the first year where you have to really get into the different kind of a grind, the second year is much less relaxed because you don't have exams. You have exam probably after 1.5 years, right?
Yes, sir. Yes,
So
sir.
you have a lot of
Sir, but
time.
even though, yes, sir, even though we are having time, we can utilize it in studying pathopharma and micro, sir. Or first we have to know some knowledge in that for doing research, because already some information is gathered after doing research by many people. So first we have to know that, then we can, I think, like that, sir. What is your...
See, this is like a wrong approach in the sense that if you think you can do research only after a lot of learning, right? And that's really
Yes.
wrong.
Yes, sir, right.
What is really, and then you will never be able to get all the knowledge to be able to do something, right? First,
Yes,
I mean,
sir, but basic.
So you can start doing research. You can start asking questions. See, that's one thing that is important to also keep in mind is that it is not that you do one project and that is the beginning and end of it. I don't think that all of research is going to be just one project in your entire MBBS period. No, you can ask small questions, right? You can play around with things that interest you, and that's essentially how you do research. It's not like you want to write a thesis after your MBBS, right? Anyway, after during your MD, you will have to write a thesis, so there is always an opportunity to do that. The idea of doing research or getting acquainted in MBBS is to get a feel for it, right? You might want to look at an small observation, right? Collect 50 information for patients. Learn how to get into the ethical committee clearances, right? You need to write a proposal in a very format, inclusion, exclusion criteria, calculating the sample numbers. So these are all things that you learn, right? And you need to learn the art of doing things, right? This is not really the first and the last problem that you will start addressing, and you will never have a point in time where you know everything to be able to ask a question. You should start asking questions. You need to get a feel of doing it, right?
Yes, sir. Sir, I have one good question from one of our team member. Like many students tend to migrate to foreign to pursue research, but what motivated you to stay in India and to pursue research?
Very good question, and my quick answer to that is very simple. If you were to stay back in India and address Indian problems, right, then it is probably, in my opinion, and this is my personal opinion, don't quote me on this, my personal opinion is that doing a PhD in India is more advantageous, right? Because if you go abroad to do a PhD, then you might not be able to answer the very pertinent local problems because those problems are not their problems, right? People work on their problems. So the diseases are different. The presentations in many cases are also different. Outcomes are also very different. And then therefore, you might actually not be able to extrapolate that directly back into the country, right? Now, having said that, this is my personal opinion. Of course, there are a lot of examples where people have gone abroad, learned a lot of very good science methodology and techniques to come back and be able to implement it, right? So it is a conscious decision that you want to make, that if you were to work on a pertinent problem, which requires a lot of technological advancements, skill advancements, yes, you should go abroad and and learn those skills and come back. But if you were to address a problem where the technology already exists, right? The patients already exist, and then you are the guy who is going to put it together based on skills that you can learn or you want to learn quickly, then probably it's a better option to stay back and pursue research.
Thank you so much, sir. And Appo F11, now you can ask your question.
Sir, these days to promote research, there is this kind of course called MD PhD integrated courses, sir. Like ICMR also recently started an MD PhD integrated courses. So can you tell in detail about those courses? What are they and how are they useful?
As far as I know, there is only one center which is offering an MD PhD course. And I'm not sure about the outcomes of this. I don't think any student has already passed through. So I'm not really sure about that. But having said that, there are also clinical PhD programs, like for example, Enhance runs a program, mostly in neurosciences. But that is a PhD program, not really a MD PhD program.
So what about foreign university MD PhD courses, sir? Like how are they?
Yeah, so foreign university MD PhD programs are good in the sense that it gives you access to both, but it is quite rigorous because you need to cramp in both your MD program as well as a PhD program into the same kind of a time scale. So yeah, so foreign universities offer this quite regularly, where you can pursue both options, where you do a PhD as well as also do an MD, right? Which is not really common in India because most universities are not very flexible to do that. But having said that, there is an MD PhD program that I have known about, but I don't think any student has actually finished that entire program yet. I might be wrong.
Okay, thank you, sir.
Okay, sir. Thank you so much. And we have another two to three few questions, sir. Like many students are interested to do research, but their college is not supporting them. So how do they approach someone to do research? And the requirement of ethical institutional ethical clearance for the studies. So will they can they do the research without taking IEC clearance from them, like on basic studies, knowledge attitude practice studies, without much clinical involvement or implication like that?
Yeah, so if you were to ever do a study on patients, it could be as simple as a simple questionnaire. It could be as simple as observational studies. It could be as simple as looking at the pathology slides. If you're going to work on patients and collect clinical information, then in any format, then you will need an ethical clearance. But of course, you could do studies which doesn't require clinical samples, which doesn't require ethical clearance. Best examples would be you could go out and ask this question, right? Of all the patients who come with a particular kind of fever during an epidemic, can I plot them on the map, right? From news sources or public domain resources. We are not working on clinical samples. We are working on data that is already public domain, right? You can plot it. You can do a lot of interesting analysis on that, which is still clinical data, but not on patients. You're not running a questionnaire that doesn't require an ethical clearance. So you can still work on a lot of public domain data sets, information, right? And that doesn't require an ethical clearance. You could work on, say, for example, if you step into microbiology, they would do blood cultures left, right, and center, right? If you do a subculture without the clinical information, and probably, for example, look at a particular mutation that causes drug resistance, that doesn't require an ethical clearance because you're not working on patient samples, you're working on discarded samples. There are a lot of information that you can collect based on samples that are discarded, right, after doing the test, where there is no clinical information tapped onto it. You don't really require an ethical clearance. So not getting an ethical clearance is not the reason that you should not do research. right? So you need to also keep in mind that what you see as or perceive as research is probably what you need to change, right? You can ask n number of questions around you, right, which doesn't require any help, any assistance, right, but still is very interesting, right? And what I can give you is an example. There's been COVID. Everybody knows COVID is all around us, right? The data, fortunately, is available in public domain. Now, I'll give an example of a bunch of students, faculty members in a small government college in Kerala, not even medicos, have put together an app which can track the geographical location of every outbreak of COVID in a state in Kerala, and also plotted the growth of the case, growth of testing, how the different districts have progressed over time. And they have just got it published in a very respected journal, which is the Journal of Medical Informatics Association, right? So it is very possible to do very good analysis, very good data collection, very good publication, right? Without really having a lot of effort into what you really think as research is. So if you think research is all about going to patients, doing a questionnaire, doing an observation, or doing a clinical trial, that's not all of research. There's a lot more in research. And you should also be open to accepting that that is also very useful and good research that can also end up in very good publications.
Okay,
so
sir.
I can share this paper after this to one of the co-hosts, and we can share it across. It's a very good example of how you can actually use data in collection. Right? This is not one person, there are multiple people who put together the best of the skills to actually create something that is useful, and anything that is really useful and impactful, of course, will end up in a publication. So the aim is not really to publish. The aim is to do something that is really good and useful, right? Publications will follow.
Yes, sir. And we have a question that do we need ethical clearance if we collect data through Google Forms online survey?
Yeah, so if you collect data on a questionnaire and clinical information with patient identifiers, right, then you will need an ethical clearance.
So what is the difference between a population-based and a patient-based study, sir? So can population-based study is also a patient-based information are being collected or something like that?
See, one way of doing a questionnaire is, like, for example, if you are on Twitter, you can do a Twitter poll, right?
Yes.
A Twitter poll doesn't require identification of individuals. It doesn't collect the personal identification of an individual. It doesn't collect the clinical implication of a patient. Anybody can accept and participate in the poll, right, in an anonymous way. That doesn't require an ethical clearance. Okay, but if you ask this question of all cancer patients, what do they feel, right? Then you're collecting cancer patients, right? So you already have decided that you're going to collect cancer patients, right? You know who they are. You want their names and probably their clinical IDs. Then, of course, you would require an ethical clearance, right? To run a poll on Twitter, you don't need ethical clearance.
Got it, sir.
Right? Because people can anonymously join, anonymously answer your question. You don't know who answered, right? Nor can anybody else know who answered it. Hope you now understand the difference. Right? The difference is very subtle. Right? So you need to design your experiment accordingly. Right? So theoretically, you can also design a questionnaire that is applicable only to a particular set of patients. Right? Run it on Twitter, right? And still be able to do the experiment.
Yes, sir.
Yes, sir. We have one last question, sir. are there any research opportunities now during the lockdown where we can work from home or assistant as a principal investigator from home, or any other institute that can provide such opportunities to interested medical students?
It's not about medical students. Anybody is free to work from home, and there are 101 questions and enough data points that you can address sitting at your home, right? The only challenge that I see is that there are not many people who want to address questions. It's not a question of data anymore. That COVID has probably thrown out the largest amount of structured data in clinical sciences anytime, right? It's absolutely, I'll say, unprecedented, right? Even governments who would otherwise keep their data in their lockers have made it available, right? I think a lot of states, I see in Twitter, a lot of states actually put out Excel sheets, right, of patients tested positive, from which district they tested positive, right? What is the growth rate of districts like? I mean, these are all information which you can use to address very interesting questions, right? The only depth is that I don't see anybody asking questions, right? You could ask this question: did the lockdown work? Right? How do you ask a question? Did the lockdown work? The data is out there, right? Another interesting question that is very simple. You can do it on Excel. You can plot the growth of new cases, right, over time. Can you predict what is the number going to be 1 week from now? I'm not saying that this is publishable research, but I'm saying that that's interesting research. That's how you learn how to do research, right?
Yes,
can
sir.
you plot a set of values, and can you predict what is the number going to be 1 week from now? Right? Very simple. Right? You don't need statistics to do that. Of course, statistics will make you better, but these are simple things that you can do, right? Now, it is not that that is unimportant. Third, you can go to Twitter, and Twitter is actually a storehouse of information if you don't really know, because patients are on Twitter, right? There are a lot of patients, even in India, who have announced on Twitter, right, like me and you, on Twitter, that I have fever, I have back pain, I have loss of smell, right? So you can really you can pass Twitter, because Twitter has an open API. You can download this data and ask this question: what are the common symptoms of people in India who have, say, for example, COVID, right? Is there a paper on it? Answer is probably no, right? So it's not a question of anymore that can I do research, right? You can do research provided you have an inquisitive mind. That's all, right? If you think that research is all about going back to patients and collecting data from patients, then you're probably wrong. Today, I'll say, I mean, in my field, in genomics, India has close to around 2,000 odd isolates of SARS-CoV-2 genomes available in public domain. Globally, there are around 70,000, right? And it is growing at the rate, it's doubling almost every 1.5 to 2 months. So by the end of this year, we expect to have at least 8 to 10,000 isolates of SARS-CoV-2. It's all available to you, right? You can ask very interesting questions, right? You can ask where is the mutation? Why is that mutation? Now, having said that, very early on, way back in March, we started this open data collection and analysis, program, which is volunteered by a lot of people. This includes a lot of epidemiological data that we sort of collected on different states, number of beds, asking very simple questions. Like, one of the simplest questions that we asked is, which states can handle the epidemic, which states cannot handle the epidemics, right? Or what are the statistics or demographics, which states beforehand, that which states will do good and which states will do bad, right? And good and bad could be, so for example, measured in terms of mortality, right, of people who are died. It turns out that very simple numbers like number of hospital beds per population, right, the age demographics of the particular state, that each state in the country has very different age demographics, can actually predict which states do good, which states do bad. Right? This information is publicly available, but yes, somebody has to put it together, curate it, and be able to do the analysis. Other question that we asked is, can we predict today which states or which labs would give false positives or false negatives rather, right? The question that we asked is very simple. There are mutations in the genome. All RT-PCRs are based on primers and probes that bind to the genome. So if there's a mutation in that particular genome at the site where the primer binds, then probably you're going to get a false negative report, right? You can ask this question. There are 101 questions that you can ask. The only point is that you need to get out of your fixed thought that, I mean, research is only that I will go to the patient, collect data or collect a sample, and that's only what is research. No, right? The 101 questions that you can ask, and asking good questions is all about, all about the initial part of doing research. So research is nothing but asking good questions, collecting good data, and doing a proper systematic analysis. Nothing else. But you need to have a curious mind.
Hello, sir.
Yep.
Yeah, actually, I was interested in doing a psychiatry related topic with respect to COVID patients, as my hospital is a COVID hospital on the first place. But then they've told me due to some other political issues that that I wasn't giving any ethical committee approval. So how can I go forward in that?
Redesign your questionnaire, put it on Twitter.
Oh, sure, sir.
See, that's what I told you earlier, right? If
Yeah.
you go to a patient and collect a particular set of clinical information, right, then you need to have an ethical clearance.
Yeah.
Right? But if you want to put it out for everybody,
yes,
as
sir.
a poll, that
Yes.
anybody can participate,
Okay.
but design in a very particular way that the data or the questionnaire is of interest to people in a particular clinical setting.
Yeah.
Right? You can collect information which is perfectly ethical. Right? But that doesn't, that should not involve collecting personal information.
Yeah.
Right? If you don't ask for the name or the ID or the phone number or the email of the patient.
Yes, sir.
And the patient is anonymous. Like, for example, if today I put on Twitter, right, are medical students interested in research?
Yeah.
Right? Do I need an ethical clearance to do that?
No.
No. Why?
It just outfits the general interests of the students.
Absolutely. Yes, it is not the perfect ideal way that you thought about.
Yeah.
Right? But if your passion is about asking that question and getting the answer, right? That is what you would do. See, there's always this problem of perfection, right? That's a perfect way of doing things, right? Because that's a perfect way that you thought that people published, right? But that's perfectly fine. I'm not saying that that is wrong.
Yeah.
But are you interested in that publication, or are you interested in getting the answer of what you wanted to know? Right? That is the right question to ask. Right? If your answer is that I just need to get the answer of it for my self-satisfaction, right? You already know how to get the answer. Right? So science is all about that passion of getting to know before anybody else, not really about publishing that one particular paper. Of course, publications are important. I'm not saying that that's unimportant, but more important and more fun is actually asking the question and knowing more than anybody else.
Thank you, sir.
Yes, sir. And I think we can end up the session, sir, as we have already reached 1 hour 25 minutes. And you said that you will share that paper. So if you have enough time to spend with us, as you are very busy with the laboratory, you can share now, sir. We are always ready to see. Otherwise, you can share that link to us. We will share it in WhatsApp groups.
Yeah. So I will share the paper with one of you. But more importantly, what I would want to state is that what as medical students, I would think that I'll say at least 1% of these medical students, I'd say there are around 150 people who participated. So I expect at least one person. If anybody of you are interested in doing computers, right? This means that somebody of you who are interested in programming, somebody of you who are interested in just crunching data that is available in public domain, somebody of you who is interested in looking at genomes sitting on your laptop, feel free to write to me. There are more than enough problems that I can dump on you, right? There's no dearth of problems. But if you're looking for a wet lab, probably not the right time. I'm not saying no, but I'm only saying that probably not the right time, right? Our labs are typically open, and before COVID, any student who had written to us saying that we want to spend 2 weeks in your lab, we have always been welcome. What we cannot pay you is the travel. We can accommodate you for a period of time, but yeah. So if you want to really spend some time to understand what this is all about, feel free to come. We also run a program called DIPS. Two of the organizers have been participants of DIPS before. That's a very small number of students that we take every year. Typically, we used to have 2 sessions every year. But this year, because of COVID, we don't really know what is going to happen. But nevertheless, we would strive to still run the program every year. But probably this year we might have a smaller number. But that should not really have a bearing on what you want to do. Whatever you require, we will try to help. I cannot assure we will help all the time. We at this moment have restrictions because we cannot have a lot of people. We cannot have a lot of people from outside. But yeah, once the labs open up, we would do our best. That's what I can assure you. But if anybody is interested in computers, analysis, genomics, right, feel free to write to me. We have more than enough problems that we can handle.
Sir, a small question. A person who has a little bit of programming language, not in depth, like he's still trying to learn, can he also join in?
If you know how to print all odd numbers from 1 to 100, you can join.
Yeah, that's great. Thank you, sir. Yeah, thank you so much, sir.
Yeah, so that is a minimum programming that you would require, 5 lines of code.
Yes, sir.
Sir, what are your ongoing projects, sir, on future projects?
Anything to do with genomics. Whether it's pathogen, whether it's human, anything that has a genome, we are interested.
So regarding the SARS-CoV-2 genome, sir.
So yes, regarding the SARS-CoV-2 genomes, there are a lot of things that happen in the lab. One of the things that we've been doing is we have been collecting all the isolates that have been sequenced from India in trying to look at what are the variations in there, how can we classify them into clades. So one of the preprints that we have uploaded 2 months ago was on a new clade or isolate of SARS-CoV-2 in India. Not new, but it has been there before, but it has never been so large numbers in any other country. So we call it as a new clade, which is called the IE or the A3I clade. Of course, you can go to my website or you can go to BioXIP and search for it. The other thing that we tried working on was to look at variants in the primer probe binding sites to be able to understand which primers or which kits work and which kits are unlikely to work, and also design new primers and probes so that they work in almost all the times. And third, very recent that we have been working on, and there is a preprint that we uploaded yesterday, is on a high throughput approach where you can do sequencing. When we say sequencing 1536 samples in one single go, where we can do detection as well as genetic epidemiology of SARS-CoV-2. And the idea of doing this was to also extend it to all future epidemics. So any of you who would ever become microbiologists, anytime in your life, if you come across an epidemic, take your phone and call me. Right, we will be able to do genomes for them, because you can do at a throughput of 1536 samples in one single go, right? And that's a huge number. So to put numbers into perspective, India in the last 5 months have sequenced 2000 odd strains, right? Now you can sequence 1536 samples in 24 hours. Right? So that's the throughput difference. And what we showed is that it's a very sensitive approach. It's more sensitive than RT-PCR, much more sensitive than RT-PCR. And with the additional advantage that you can also look at the genetic epidemiology. So you can read this preprint out there. It is. I have also posted it on Twitter, also on my Facebook page, or you could go to the website and sort of read it. Apart from that, there are a lot of other things that we try to look in genomes and trying to look at different states at this moment. In the next couple of weeks, we should have some data on states like Andhra Pradesh, states like Kerala, where there's also a lot of influx of population from abroad, because traditionally these are states where a lot of people stay abroad and they travel back into the country. And also states like Orissa, where a lot of travel has happened between states in the country after the lockdown has been imposed. So the idea is to also be able to understand how does this dynamics work, right? If there was a strain which, for example, moved from Gujarat to Orissa because a lot of workers worked in Gujarat, and after the lockdown they moved back into Orissa, you'll be able to know what kind of strains would they bring, right? Because they got infected from Gujarat, and so they would get the prevalent strains from, or isolates or plates from Gujarat. And when they go to Orissa and then they seed the new infections, they would essentially seed the viruses that they had got in the first place. So you will be able to sort of draw maps on where did these infections come from, where did it come from abroad, what percentage of it come from abroad, what percentage came from local travel, and now be able to assess in a sort of scientific way, how did the infection spread by people traveling across states or across countries, right? And that is something that is quite interesting, of little clinical relevance, but a lot of academic interest, because now you will be able to, for example, predict if a new epidemic happens, right? If you impose a lockdown and people move from one state to another, how is the infection going to spread? Probably it's all about preparing for the next epidemic, right? And we can sort of advise people to say what we should do and what we should not do.
Yes, sir. Thank you so much, sir. So, Madhav sir, I think now we can end up this session. So, after taking 2 to 3 formal pictures, I think we can end up the session.
I have actually taken all the screenshots during the session.
With our team.
Yeah.
Like our team does.
Yeah.
Like I request team SMA to switch on their camera. Aisha, the remaining participants, you also
Yes,
please switch on your cameras, because I will take a screenshot of all 3. How many participants are there?
Sir, there was one more interesting question that, during the camera session, we can actually have it. That is, if he doesn't have a programming, still wants to learn about analysis and statistics and want to do, still can we approach you, sir? Was a question.
Absolutely. So one of the things that we are currently going to run is a course on how to analyze and interpret genetic variants, right? You would see a paper which says that a new gene or a new variant for this particular disorder has been identified, right? How do you interpret that variant? How do you scientifically evaluate that variant, right? Or somebody says that, somebody wrote a paper saying that I have found a new variant for, say, Wilson's disease. right? How do you evaluate that particular piece of evidence in a very systematic way? It's a very laborious way, actually, very systematic way, by using a set of guidelines and tools to be able to say that, yes, this individual was right, and according to my independent assessment, this is a pathogenic variant, or my independent assessment, this is actually crap. Right? How do you go about doing that? In a very systematic way. You don't require a lot of programming skills, but you need to have some great interest in genetics, because it requires a lot of patience and a lot of your time, effort to be able to do that. So we will be running this course probably starting early next month, and I can of course circulate it across you. But the caveat is that only people who are interested in doing something should join. There are no certificates to be provided, right? The only
So is it
thing
an
that,
online course?
yeah, it's an online course.
Yes, sir.
Many students are asking even certificates for this webinar, sir.
You should not give
So
certificates.
that they can mention in
They should mention in the CV, right?
yes. Exactly.
Sir, is there any free fee for this course, sir, which you have mentioned?
No. It's completely free.
Yes, sir. Thank you, sir. I thank you so much for taking your time and on your busy schedule. Even though the session was planned for only 1 hour, we got so many questions that it has taken almost close to 1 hour 40 minutes. I'm so sorry for that, taking such a long session. But you have answered almost every question that we have asked you, and I hope many people would be approaching you either for the course or interested in working with me, and maybe I will also write an email to you in a due time. And yes, sir, and please excuse us if if we have not organized the session well or not met your expectations.
Yes,
That's
sir.
it, sir.
No, absolutely.
And,
I enjoyed the session, and yes, yeah, anybody could write to me directly. My email is anyway available, or you can share my email. So if I get delayed in responding, that doesn't mean that I'm not interested. Just bear with me. But I would surely respond to you.
You are really very
I
fast
want
in
to add
responding,
last
sir.
point that Sir is very, very kind person. I have no words to express his kindness. So thank you so much, sir, for accepting our invitation. And I am looking forward, and I hope I will work with you definitely in future, because I want to pursue research as my career. Thank you so much, sir.
Thank you.
Thank you, sir.
Bye, everybody.
Bye.
Bye, sir.
Okay, I thank you so much everyone for all the participants for joining in the session.
Thank you so much, everyone, for attending this session by giving your valuable time.
I request actually Team SMR to kindly stay back, and rest of the people can actually leave the session.
Could I get Sal's email ID once?
Yeah, actually we'll share that in the group.
Okay, thanks a lot.
Yeah.
Can I get the link for the YouTube video of this session?
Yeah, we will also share it on the group.
Actually, we haven't put it up online. It is not online yet. Yeah, there was some disturbance with
YouTube video
Live.
they're asking.
Yeah, YouTube Live, there was some disturbance so that we couldn't make it happen. But we are going to definitely put it once the session is completed.
Okay, thank you.
Mother, stop the recording and save it. You can stop the recording here.